Hi, I'm Jay Phelan. Let's learn some biology. When we think about heredity, rightly, we credit Gregor Mendel with helping us to understand some things that had long been unclear to many people, including Charles Darwin. And Mendel discovered many things, figured out many things. We summarize probably his two most important findings in what we call Mendel's laws. And we're going to talk now about Mendel's law of independent assortment. And previously, we've discussed the more important one, which is Mendel's law of segregation. So we won't spend time on that today, other than to say that it points out that for a diploid species, you have two copies of every allele. But when you make a gamete, you only put one of those into them. Then at fertilization, that gamete with one allele comes together with another gamete with one allele, and you restore the diploid number again. That's how the offspring can have some traits from the father, some from the mother. It's how they can avoid having an increase in the amount of genetic material every generation, and so on. So that's the important rule, Mendel's law of segregation, and it runs through all of genetics. Mendel studied peas, but it's important to remember that genetics refers to inheritance in any species. We don't have to look at peas. They were good for him, but we can look at a variety of different organisms if we want to study them. And I'll first think about something like color in a giraffe. So we have these white giraffes. They're albinos. They have a homozygous recessive condition for this pigment-producing gene. And if you were to cross a giraffe that has the homozygous recessive condition, we'll say two little m's for melanin, with a giraffe that is normally pigmented and is true breeding, meaning that they'd always produce the same one. So we know that it is big M, because of Mendel's law of segregation. The mother would always contribute the little m, father would always contribute the big M. They'd all be heterozygous, and because the melanin, the pigment allele, is dominant to the recessive albino pigment, they would all be normally pigmented. So that's it. So they'd all look the same. We could take these, though, these heterozygous individuals that have one big M, one little m, and we could do a cross of those. So now you have to remember Mendel's law of segregation. Half the gametes the mother produces are going to be big M, half are going to be little m. Same thing for the father. So when they come together, we don't know what's going to happen, what the outcome's going to be. We use our Punnett square to serve as a bookkeeping device that says these are all the possibilities. If we had an infinite number of giraffes, we'd get 25% each of these. So when we look at these, if we did this cross, we'd find that the outcome is that 3/4 of them are going to show up with the dominant trait. They either get two copies of it, one from the mother, one from the father, or they get one from the mother and then the recessive from the father, or one dominant from the father, and so on, whatever. 1 out of 4 is going to show the recessive trait. It has to get the recessive allele from the father and the recessive allele from the mother. So looking at a single gene trait, that's just how we'd analyze it with the Punnett square. But you look at a giraffe, it's not just one trait. It's not albino, normally pigmented. It's got a neck. How long is the neck? It's got eyes. What color are the eyes? It's got a foot. What shape is the foot? It has a lot of different traits. And we don't necessarily want to only look at one trait at a time. So we can look at multiple traits. For instance, in cats, we could look at the gene that influences the fur length. Is it a long-haired cat? Is it a short-haired cat? We can look at their coat color. Is it white? Is it darker? Is it non-white? And it turns out for these, actually true, that the short hair allele is dominant to the long hair allele, so we'll call that big L. The long hair allele is recessive, we'll call that little l. We look here then at the long-haired cat, and we can see that, okay, it shows long hair. It's a recessive trait. It's got to have two copies of the recessive allele. We know its genotype. We look at the short-haired one. We know it has to have a dominant allele. Maybe one, maybe two. We don't know. So we'll say it's big L, or big L, little l. We just leave it at that. We're not sure. For coat color, then white fur is the dominant trait. So we'll give it a capital W, and the dark fur is a small w. So we look at the white cat. We see, all right, it's got at least one big W. It might be big W, little w, or it might be big W, big W. The dark furred cat has the recessive trait, has to have 2 copies of the little one. So now we know these. These are just different traits that we could look at, but we could track them at the same time. We don't have to just look at one trait at the same time. So what if we took a cat that was heterozygous for fur length as well as color? And we look at them at the same time. So if you have a cat that is doubly heterozygous, we have to think about what's in the gamete, because when we're going to make our Punnett square, remember, we put the female gametes on the top, male gametes on the side, and then we figure out, what if these came together, what if these came together, and so on. So to do that, if it's doubly heterozygous, the parent's genotype is going to be big L, little l, big W, little w. It would show the dominant trait for each of these traits. Now the gametes, people mess this up a lot because they're like, oh yeah, it's going to have 4 different kinds of gametes, a big L, a big W, a little w. But that's not right, because remember, a gamete has to have 1 copy of every trait. So it can't have just a big L and then nothing for the W. It has to have something for the length of the fur, something for the color of the fur. So it looks like this. Maybe it's a big L and a big W. But it might be the big L and the little w. We don't know. Mendel's law of segregation. Might be the little l that it gets with the big W. It might be the little l with the little w. So all of those are equally likely. That individual is going to produce equal numbers of each type of gamete there. So we could take 2 different cats that are both doubly heterozygous, and we could cross them now. So previously we did the cross of the normally pigmented but heterozygous giraffes. Now we say, let's look at cats that are heterozygous for both and cross them, see what we get. We make a Punnett square, but now it's bigger. Instead of 2 by 2, it's 4 by 4. We list, just as we did previously, all the gametes that the mother produces across the top. There's our big L, big W, our big L, little w, and so on. Same thing for the father. Then what you do is you go through and you fill in all the squares, 16 different possible outcomes. You don't know which one you're going to get. Chance plays a big role in genetics. If that big L, big W gamete, the egg of the female, is fertilized by a sperm that has a big L, big W, then that diploid genotype is going to be big L, big L, big W, big W. We can figure out then it's going to show the dominant trait for both of them. Or it might be that one. So the little l, little w gamete from the female gets fertilized by the big L, big W, and that's going to be big L, little l, big W, little w. And again, it's going to show the dominant trait for both, but it's got a different genotype. You go through and you solve all these. Maybe it's the little l, big W sperm that fertilizes this egg of the female. Again, it would be doubly heterozygous. Again, it's going to show the dominant trait for both. Or it might be the recessive allele for both in the gamete and the recessive allele for both in the other gamete. They come together. Now it's going to show the recessive phenotype for both traits. So you look at all these different possible outcomes, fill them in. I've got the genotype written in, and then I have the phenotype. If you look, it's either dark gray color or white. It's either got the long shaggy fur or it has the short fur. From that, we can analyze what the outcomes are. We could say, out of these 16 possible outcomes, how many of them are white versus how many are not white? And if you look at them, I count the first row, 1, 2, 3, 4. Second row, 5, 6. Third row, 7, 8, 9, 10. Last row, 11, 12. 12 out of the 16 have white fur. That's 3/4. What percentage have the dark fur? Well, you look, there's 4 out of the 16. 1/4. Now we turn our attention to the other trait. The long hair versus the short hair. What percentage have short hair? Well, you look, you can count them all up. You'll see in the lower quadrant, those 4 are long-haired. So 12 out of 16 have the short hair. When you look for the long hair, it's 4 out of 16, 1/4. And so you see the same pattern that we saw when we were looking at just one trait at a time. This is another way that you could organize the outcome from the cross. So at the top, I'm just looking at short versus long hair. And you see that 3/4 were short-haired. And those short-haired ones, they're showing the dominant trait. They might be homozygous dominant, or they might be heterozygous. In fact, we'd see 1/4 of them would be homozygous, half of them would be heterozygous. But 3/4 still show the short-hair trait. 1/4 shows long hair. If we look at white versus non-white, we're going to see that 3/4 were white-furred, but 1/4 of those were homozygous dominant, and half of them were heterozygous. And so The way Mendel described this when he looked at multiple characters simultaneously, he phrased it this way. He said, the relation of each pair of different characters in a hybrid union is independent of the other differences in the two original parental stocks. In other words, that's kind of a complicated way, but he's saying that what you inherit for one trait doesn't influence what you inherit for another trait. The traits assort into the next generation independent of one another. So that's an important take-home message. That is Mendel's law of independent assortment. The allele you pass on for one trait has no effect on which allele you pass on for another trait. If you go through and you look at those 16 possible outcomes in the Punnett square, and you can look at what's the frequency just of one trait. In other words, you ignore one of the traits, and what you're really looking at is a 2x2 Punnett square for one trait. So doesn't matter what the other trait is doing. You end up still with that same cross. It's interesting to note that Mendel saw this, and he concluded that what you inherit for one trait doesn't influence what you inherit for another one, which is correct. But his understanding of the process, the underlying process, actually wasn't correct. He didn't have access to good microscopes, so he couldn't see what was going on. He didn't know that chromosomes existed. So he imagined that every trait was this hereditary factor just floating around. So you have two copies, and one goes to one gamete, one goes to the other. So of course the traits don't influence each other, because they're all just free-floating. Why would they? In fact, though, that's not how the traits are carried. They're on chromosomes. And you don't even have to know this to realize that when you look at some traits, you see, well, how come people with red hair, frequently that seems to be associated with having more freckles than usual, or not having red hair, not having freckles. Seems like those traits, what you inherit for one, influences what you inherit for the other. And it's true. And there are pairs of traits for which that is true. Now, why is that true? Well, think about it. How many traits make up a human? I've got hair color, hair texture, skin color, how far apart my eyes are, and so on. 100,000 traits, probably more. 23 chromosomes, 23 pairs of chromosomes. So those 100,000 traits have to be all put on the same chromosomes. And when we look at how the gametes are being produced in meiosis, you realize that you've got the maternal chromosome and the paternal, and they're going to go to opposite places. But all those traits that are stuck together on the chromosome, they kind of get carried along together. So we say they're linked. We're going to talk more about this later on in the course. But because of chromosomes, some traits just have to be. We can't have them all freely floating around. Now, how did Gregor Mendel miss this? He was studying his peas. They have chromosomes. He studied 7 different traits: seed form, seed color, the pod color and form, the seed coat color, and so on. These 7 different traits. And just through dumb luck, it seems like all the traits he studied were on different chromosomes. So when they're on different chromosomes, they follow his law of independent assortment perfectly. We say serendipitously. Maybe he studied more traits and he only reported these because he understood the results. Not really clear, but we'll see that by finding out situations where the law of independent assortment is violated. By looking at the extent to which it's violated, it's going to give us some insights into how far apart the genes are in the chromosome and various other things, but we'll look at that later on. For now, we'll stop and say Mendel's law of independent assortment, also known as his second law, or in my opinion, his lesser important law.